Hello, everyone. Thank you for joining us today. It's definitely exciting to see a number of familiar names on the webinar. I just want to say thank you. I know this is a very challenging time, in sciences right now with the ongoing pandemic, and everybody's working from home. But I just want to say, if anyone needs any assistance, feel free to contact us anytime. DNASTAR is still open. We're based in Madison, Wisconsin. Wisconsin currently has a stay-at-home order, but DNASTAR is up and running and fully operational. As Sharon mentioned, my name is Brian Anderson. I've been with DNASTAR for just over 15 years. I went to undergrad and graduate school at the University of Wisconsin-Madison. Personally speaking, I enjoy wilderness camping, canoeing. I'm a big fan of northern Minnesota. The Boundary Waters canoe area was wilderness. Also highly involved with, poverty awareness advocacy, volunteering. I'm a state of Wisconsin ombudsman for a local nursing home. I went to Capitol Hill to speak to some of our constituents in regards to poverty-related issues. So I'm really excited to be able to discuss, some of the solutions that we have in our software. In those 15 years that I've been with DNASTAR, these are some of the notable projects that I've assisted with. By far, one of the most interesting was helping the Egyptian Museum in Cairo track the lineage of King Tut that was eventually published in National Geographic. I have a very close connection with Ethiopia. So about 3 or 4 years ago, I had the privilege to actually go visit and talk with the previous president of Ethiopia and also provide some solutions for Addis Ababa University, the Institute of Biotechnology. And we have a number of different researchers who are working with viruses, Ebola, coronavirus, and it's been a pleasure to be able to assist those researchers. As Sharon mentioned, the focus of today's webinar will be specific to SeqBuilder Pro. SeqBuilder Pro is a foundational tool in our what we call our molecular biology suite. So the molecular biology suite would be the configuration that would provide you with solutions for sequence, protein editing, automated annotation, primer design, in silico cloning. There's also some other tools in that suite that would allow you to do Sanger alignments. So SeqMen Ultra, or if you're more familiar with our SeqMen Pro legacy application, pairwise and multiple sequence alignments with our Megaline Pro application, genome to genome alignments, blasting, as well as a number of other workflows that we support in that suite. Today's overview will consist of, a general overview of the look and feel of SeqBuilder. Hopefully, whether you are a long-time user or you may be new to the software, hopefully this webinar provides you with some value. Maybe you find something in here that you may not be aware of. The focus of our software, though, is to assist researchers in trying to make their research easier. So followed by that general overview of the look and feel of SeqBuilder, we'll do some automated cloning. Specifically, we will insert a target into a p-enter vector, followed by a gateway expression cloning insertion using the recombination elements that are present in that p-enter vector. Followed by that, we'll do some primer design, PCR primer design, followed by some sequencing primers in our SeqBuilder Pro app. We'll walk through some codon optimization. So we'll take a protein, back translate it in a highly expressed, E. coli codon usage chart. And then we'll do a demo of our auto annotation capabilities in our SeqBuilder Pro. So there's a feature library that's built into SeqBuilder Pro. Not a lot of people realize it, that you can use it for plasmid auto annotation all the way up to bacterial genome auto annotation. Probably in the 4 to 5 megabase E. coli size would be the largest projects that people have auto annotated with SeqBuilder Pro. To conclude, we'll have a question and answer period. So if you have any questions, make sure that you can send them to Sharon via the question tab. I will also take breaks along the way between the different topics to answer any questions that might come up. So let's jump into the software. When you initially install Lasergene, whether via demo or you might just purchase a software, you might be a new install, what's going to automatically pop up is what we call our DNASTAR Navigator interface. As you can see here, the Navigator is organized either by application. So here's all the different applications that are in our Lasergene suite or DNASTAR Lasergene suite. So whether you need tools for basic mold bio, tools for next-gen sequencing, microarray analysis, RNA-seq, ChIP-seq visualization, looking at Venn diagrams, heat maps, scatter plots, etc. Or we also have a workflow-focused organization that will point you towards the application that's suitable for whatever workflow that you're using. Say, for example, here, if you are interested in agarose gel simulation, I would simply just click on agarose gel simulation. And then what's automatically going to pop up is our SeqBuilder Pro application, where you would add your specific nucleotide of interest and specify your enzymes, primers, and all the other markers that are associated with agarose gel simulation. Also in the interface is the capability to have direct links to resources that we have created online. So to the right of automated cloning here, there's a little I for information. If I click on that, it's automatically going to take you to our websites. And sometimes in this webinar, our website, it slows down a little bit, but once it does open, as you can see, there's a workflow page. As I scroll down the workflow page, it will actually provide you some information, stepwise information on how to do automated cloning within the software, with direct links to resources, tutorials, FAQs, citations of people who have cited our software, as well as user guide information. If I click on Organize by Application and go back to the SeqBuilder here, if I click on I next to SeqBuilder, this will directly take me to the SeqBuilder Pro landing page. We've taken a lot of effort to put as much information in on the website specific to different parameters, workflows that are present in the software. And one thing that might be helpful if you're new to the software is in the top left-hand corner here under SeqBuilder Pro tutorials. You can actually go through stepwise tutorials on how to create and modify primers for TA cloning, etc. So the focus of our navigators is to very quickly point people to the correct tools within our suite of applications that we provide, as well as what I like to prefer and use this tool as a quick interface into our solutions. So say, for example, if I right click on the icon on my taskbar, I can actually pin this to my taskbar. And instead of having 7, 8 different shortcuts on your desktop, I basically have a quick entry point in the application that I can use to navigate to my application of interest. With that said, let's just simply open the SeqBuilder Pro application. When you initially open SeqBuilder Pro, you will get a very basic window like this if you launch it from the application window. If you launch it from the other different workflow windows, it will actually open up, the workflow of interest. So for example, gel electrophoresis, what we did previously. In regards to my computer here, I'm running a Windows 10 laptop with roughly 16 gigs of RAM. So by default, this background window will pop up. If you're on an Apple or a Mac computer, what you're going to see at the top of the screen is simply the file edit features, enzyme cloning, etc. Background that is common in Mac applications. To add your data to the software, there's a number of different ways you can kind of get data into the application. One way that I preferably use is what we call I drag a file into the application. So for example, I have a folder of data here. If I simply just want to take this sequence and drag it into my project, it will automatically open that project and the file would be annotated. This is a sequence from cow. This is something that we pulled down from GenBank, and since there's annotations, it will automatically populate those annotations. You can visualize those in the feature tab that we'll talk about later. I'm going to close this out here. Other ways you can kind of get data into the software, I can also go bring in something via Entree. So for example, from the file pull down, I can open an Entree sequence. In this random case, I'll just type in 1, 2, 3, 4. And what this will do is we'll connect to NCBI. It will ask you, okay, do you want to download the data? Specify the location you want to download it. You hit save. You want to replace it? Yes. And then it will pull in that data from NCBI, all the annotations, citations, comments that are attributed with that file. In this specific case, let's see what this is. Comments, it's a random gene from sheep, like so. Right. Another way that you can kind of bring data into the software electronically through NCBI would be under the net search pull down. So if I go net search, new text search, so built into the software, you have the capability to search through NCBI, whether by a specific author or sample or breed or strain or all searchable fields. So a popular protein that people are searching through right now is the S protein from coronavirus. So if I simply just want to search for that protein, what's going to pop up is a search window. This is also similar to our BLAST window. When we submit a BLAST through SeqBuilder Pro, there's BLAST results that come back. Here, I can actually double click on any of these selections. It's going to automatically open the website where that nucleotide or protein is represented. So in this specific case, if I wanted to bring in this furin from human, I can simply just check this. It gives me the option here at the top where I can open it in a specific application. I can also do multiple selections and checks. I can do a batch save, and that will actually essentially create a multiple sequence file on whatever folder that you saved this data to. Or in this specific case, I will simply just open this up in SeqBuilder Pro. And it's going to ask me where I want to save the data to. I'm going to simply just use the defaults and select OK. And then what I've done is brought the data in via another way into the software. Once the data, is in the software, there's a number of different views, toolbars, pull downs, and panels that we have created here. So in the bottom portion here, we have these various tabs where you can kind of toggle between the circular view, linear view, sequence view, primer design views. There's also some more secondary views. So if you're doing primer design and you want your primer saved, it's going to automatically save to a primer tab. So if we pull this down from NCBI and you want to visualize annotations, if I click on the comment tab, there'll be annotations, any publications that might be attributed to this file. As I scroll down, you can kind of see the information that's available here. There's also site summaries in regards to mini maps and site summaries of restriction enzymes. So by default in the software, there's single cutters that are being shown of restriction enzymes. So those single cutters would be represented in the mini map view as well as a site summary. You can also look for absent sites. So say, for example, you want to visualize all restrictions enzymes in the rebase catalog and find that unique restriction enzyme that doesn't cut your target. That will actually display here in the site summary view where you can find those absent sites that you can use for downstream cloning workflows. What I'm going to do here is I'm going to simply click on the feature view and just kind of talk about the feature view a little bit. Within the feature view, there's a number of different annotations that you can add or create. Any manual annotations that you add to the software under the feature pull down, when I highlight a selection and create a new annotation, would automatically be populated here. Within these feature annotations, you can kind of change the strand, you can change the feature, the nomenclature, the color, the style, etc. very readily and easily. The next panel that we're going to kind of take a look at here on the top right-hand view is basically what we call the style panel. So I'm going to click on the circular view here in this specific case. So say I wanted to change this color here from an orange of this furin, which is represented as a fill orange here, and I want to change it to pink. What I would simply do is click on or highlight that selected feature, click on that fill button, click on the color that you want to change it to. You can also define custom colors, hit OK, and that color is going to be readily changed. You can also change the font, the style, of selected items, all features, all enzymes, sequence, circular, title, rulers. There's a number of different options you can kind of change in regards to what fonts you want to utilize that are built into the software. You can actually make this as busy or as simple as you feel, which we'll kind of talk about here in a second. The next tab here would be the enzyme tab. We'll actually kind of use this a little later in our restriction enzyme cloning workflow. But say you're looking for a specific enzyme of interest. So kind of we'll talk about this in a second, is say I'm looking for unique or absent sites via the restriction enzyme tool here, but maybe there is another enzyme that I'm looking for. So for example, NCO1. Then you can apply or look for directly NCO1 and see whether that cuts your enzyme of your target of interest, like so. The other tabs here are more accessory type of tabs that are specific to different, tabs that are open and present, in the software. So for example, when I have the linear or the circular view, the circular map view here, I can actually change, what is being displayed on that plasmid, whether the title, the length. When I'm in the primer design view, whether I want to see, alternate pairs or priming sites. And then as I scroll down here to the bottom, this would be the location where any partial translations would be applied to, and names, whether you need to join partial translations, would be populated down here. The final tab we'll talk about here is the details tab. In the details tab, this would actually be specific to whatever is selected in the various views that you toggle on. So for example, in this specific case, we highlighted and changed the color of furin. So it's going to show me all the specific attributes or annotations that are, specific to that gene, as well as it's going to give me some statistics, whether ACGT breakdown, the charge at pH 7, the isoelectric point, etc. Right. Now let's get into the various tools that are present in the software here on the left-hand side. I'm going to click on the sequence view. And then there's a number of different tools here. Some of them are more specific for layouts. The two that we're going to focus on today would be actually the Enzymes tab or tool, and then the ribosome or the translation and ORFs tool. If I click on Enzymes, what is being shown is actually unique selectors that either I have created in the software or have already been present in the software. So selectors such as finding blunt cutters, 5' overhangs, unique or absent sites. I've also created more unique selectors that are specific to different projects I was working on. So CPG methylation blocked or impaired by overlapping CG. So anything that you see here is actually something that you can modify, edit, or delete, or create within our Enzyme Selector Manager. So if I go to Enzyme Selector Manager, what's going to pop up is the various selectors that are created and present, in that tool that we just clicked on. So say, for example, you're working with a specific vendor's catalog of restriction enzymes, and that's all you use. So in here, I can actually create a new selector, and then if I go to type, I can actually pick just those enzymes that are available to me that I have at my disposal. So maybe you can call it company 1, and then you would save it out as okay. Once you click on that tool, that company 1 will be present in that selector that you can use for downstream analysis. Okay. The next tool that we're going to click on here is what we call our translations and ORF. Once I click on this, if I click on reading frames, this will actually show me the top strand or bottom strand or all strands or all different frames, translations, and those translations will be populated to whatever view that I'm visualizing. The next functionality here is open reading frames. So say, for example, you want to look for potential open reading frames of interest within your nucleotide. In this specific case, if I click on ORFs, top frames, this will show me the open reading frames based on the default open reading frame detection options that are set, in the software. If you wanted to change those open reading frames, that is possible. So under the sequence pull down, there's an ORF detection option selection. So in here, we give you a minimum ORF size. So by default, 25 amino acids is set. So maybe you want to look for amino acids, open reading frames of at least 100 amino acids, and showing only open reading frames that have a start codon. I would simply change it to 100, show only ORFs with a start codon, hit OK, and then what's going to be shown is just those open reading frames that fit that criteria. So if I click to the linear view here again, and then if we focus on this furin, if I click on this little star, shows me the full-length translation open reading frame of this specific gene of interest. As I mentioned previously, I'm going to click on sequence here again. There's also accessory tools that are present down here, whether you want to very quickly go to a primer design layout, a full sequence layout. Maybe you've manipulated this, this view, you have the flexibility to drag tabs. If I don't want this feature view to be at the bottom, I would prefer more of a sequence view at the bottom. You have the capability to drag this view to the bottom and have a linear view at the top and a sequence view at the bottom. You can customize this to your heart's content. Say you get, you want to go back to the defaults, you can actually reset the default layout view, or maybe you're coming over from another application. So we're getting a lot of customers right now that are transitioning from vector NTI, say for example, and you've created a vector NTI layout view specific to something that you're familiar with. Within the software, you can actually save those default layout views. You can save those colors, the styles, the fonts directly from our view pull down. There's an option to actually save a layout as a default template. You can also reset accordingly, restore your default layout, or maybe you have a specific layout for a unique project and you want to look at just the open reading frames in a given project. You can also apply specific views specific to whatever project that you're working with. For some of our long-term customers, as Sharon mentioned, we do also have what we call, a legacy application called EditSeq to help some of our customers transition over from EditSeq to SeqBuilder. We also have a compact EditSeq layout view. So if I click on this, and you probably want to turn off some of the different open reading frames that we apply to the sequence, that will give you your classic EditSeq view. Okay. At this time, the other options that we have, in this overview or feel, this view would be basically the various pull downs that are present. For the sake of time, I'm not going to go through every specific pull down. I'm assuming that, people are familiar with the common things that are present in a file pull down, how to print, how to save, how to edit, how to copy, how to search. There's search capabilities where if I want to look and search or find for a specific nucleotide string or an annotation, those are all present in the file edit pull downs. Similar, the view capabilities, you have the ability to show hide panels, remove splits, reset default layouts, and etc. We already mentioned the enzyme pull down, so this is where you're going to go if you want to create your unique selectors, where you maybe want to open the enzyme manager and add your own unique enzymes. You can actually create those in the enzyme pull down. The sequence pull down, this is where you want to go if maybe you highlighted a selection, you want to do a new sequence and translate that nucleotide of interest. Or maybe you got a vector map from a vendor and you need to reverse complement or change the origin replication to translate or change that protein in some way or change the origin directly. The cloning pull down, this will be the direct links to the various in silico cloning methods that we have included in the software. So today we're going to basically do the PCR directed restriction cloning, which is essentially just point and click. And then once we've created that restriction clone, we will do a gateway expression clone directly from that expression clone. There's also in silico cloning wizards for Gibson cloning and fusion, GeneArt, TA cloning, directional topo, as well as more of a manual approach. So if you simply just want to amplify or add your enzyme of interest using some of our PCR primer design options and add those restriction sites 5' and 3' end, you can find that corresponding enzymes in your expression clone and use the copy restriction insert and paste restriction insert functionality. The primer design pull down, this is where you're going to go towards if you simply just want to highlight a selection and create a random forward or reverse primer, or if you simply just want to use the create primer pair functionality that exists in the software. Like all of our different applications, we also have the capability to blast a selection. So I can highlight a nucleotide or a protein and simply just blast a selection. It's going to automatically connect to NCBI. You can specify what program you want to utilize as well as what database you want to blast and pull data down from or search for similarity. Okay. With that said, that's kind of the general overview of the look and feel, of SeqBuilder Pro. Is there any questions at this time that I can answer?
Yeah, thanks, Brian. We've had questions coming in like crazy, most of which I've been able to answer by sending people to a particular help page. So I'm just going to ask a sampling of them. The one that's come in just now that I have not answered is there a way to save a simple multi-FASTA file using SeqBuilder Pro? On the same realm, if we have a genome opened, is there a way to export individual multi-FASTA DNA annotations or proteins?
Thank you, Sharon. Those are great questions. So the first question is, within the software, there's what we call projects. So one of the things under the file pull down is you have the capability to create, new cloning projects. So a new cloning project is going to be automatically created as we do the in silico cloning lizards. There's also the option to create a new project here. So file new project, which would be different than a cloning project. So in this project, you actually have the option to import, data. So for example, if I wanted to import a multi-selection or and simply drag that into a project, it will actually pull that data into the project. From there, I can highlight a selection and then actually right click and actually export that data back out. So one easy way is if you're doing it via a project, you can export it and it will export out as a multiple sequence file. The other option is I didn't really talk about in the tools pull down here at the top, but there's actually some more automated bioinformatic type of workflows that we kind of support. So this is actually, I'm showing you the coming release of 17.1. You'll have the capability to batch clone. So say, for example, you have 96 targets and you want to insert them into one expression vector, but have 96 amplified targets, inserted into that clone. So you have the capability to automate some of those. So one of those is I can export all those sequences in a project, as a single file. To your second question, okay, you can actually bring in genome information into SeqBuilder Pro. I would say the largest chromosome that we brought in is, a human chromosome. So it's going to take things a little bit longer when you open projects of that magnitude or that size. But for E. coli or tuberculosis, you can very easily bring those very large genomes into the software. But what you can kind of do is then you can actually extract features as sequences. So one of the options here under the tool pull down is the capability to extract features as sequences. So what you would do is you'd bring in your genome of interest and then basically utilize the extract features as sequence functionality. You can specify the type of features. You can actually go even further to specify the name of a given feature. So for example, what I've done is I've brought coronavirus genomes, into the software, created them, put them in a project, highlighted a selection, and then extract just the S protein from 96 different coronavirus genomes. And then you can actually focus on just on those genes of interest that are present across those genomes that you're further evaluating.
That was a great question, and thanks for answering that. Those new items in the tools menu are things that never existed in our old legacy applications. These are new for SeqBuilder Pro, and I don't think that a lot of people realize that they exist, and they are such powerful tools for doing work in big batches, and they can save you a lot of time. We have a couple more questions. One is, can you connect to a local BLAST server?
That's a great question. Built into the software, there's not really a local BLAST database per se, but we have assisted a number of different companies, pharmaceutical companies, to be able to integrate local BLAST solutions, but that would be a case-by-case basis. So currently, out of the box, BLAST functionality would be through NCBI, but depending on the location, there might be a way in which we can assess a local BLAST of some sort.
Great. And then can you use SeqBuilder Pro to find gaps in a sequence?
Great question. So under, I'm going to just close this out here just to minimize some of the things that are open. Under the file pull down, you have the option, under the edit pull down, you have the option to find things. So if I go edit find, what's going to pop up is our find interface, and it's hiding right now on my screen somewhere. Sorry, I have to close some of these windows. Oh, there it is. It's on a different monitor. So what you can kind of do is you can actually search for literal strings, but the second option here is you can actually allow ambiguity or search for ambiguous information. There's also the capability to look for open reading frames, restriction sites, specific features across whether it's a genome that you're bringing in to the application or simply a plasmid. So yes, the answer is you would simply type in your nucleotide string here and then specify allow ambiguity, and then it would search and find that specific string of interest.
All right, I think that's it for the questions related to this part of the webinar. So just you can move on ahead to the next thing that you're going to cover, and I will continue taking questions to ask at appropriate places along the way.
Okay, cool. What we're going to do here is once SeqBuilder is open, I'm just going to close out some of these projects so we're not confused by some of these that are open. I'm not going to save this. Oh, there we go. What we're going to do here is we're simply just going to open this specific sequence. This specific sequence is a gene from cow. I'll open on my other screen here. I'm going to maximize the window. As you can see, when it opened, all the default color styles are maintained. I'm going to actually click on the sequence view, and then simply I'm just going to highlight this specific gene of interest. What we're going to do here is we're going to actually insert this into a p-enter4 vector from Vitrogen. Couple of things just to think about, as you start your cloning experiment. It might be helpful to turn open reading frames on. So under the ribosome palette tool, what I typically like to do is I will show my top frames. So that way you can kind of maintain or ensure that whatever cloning strategy that you're utilizing, that the frame, the open reading frame is being maintained across the experiment. So what we're going to do here is we simply just highlight that selection. So a couple things within the software, I can randomly highlight selections. Everything that you see here is something that you can edit. So say, for example, I wanted to copy paste a specific tag of interest, into this vector. Everything you can kind of see here is something that is truly, editable. As what I just did here is I typed in some random ends, 5' of these initiated methionine. But just to clarify, everything you see here is something that you can edit. I can also take a selection, I can copy it. What I'm doing here is I'm using Control-C, and then say if I wanted to paste it to a new project, I can open a new project, and click Control-V, and that piece of information will be automatically copied into that new project. With that said, what we're going to do is we're simply just going to click on this feature, this gene from cow, and then we're going to simply just insert it into a p-enter4 vector. So to do so, what I'm going to do, highlight my region of interest. Whatever view that you're in, whether the circular, linear, sequence view, really doesn't matter. I just prefer the sequence view. The next thing I'm going to do is going to go to the cloning pull down, and then we're going to select PCR directed restriction cloning. What automatically pops up is kind of mentioned before is what we call our cloning folder per se. So in this project, there's essentially there's a project tab, there's a clone tab, and then there's a summary tab. In the project tab, it keeps track of your inserts. So there's my starting insert that's populated, vectors, as well as resulting clone. In the cloning tab, you have the option to save your experiment. So in this specific case, the experiment will be saved out as restriction 1. I can also toggle between different methods. So in this specific case, we selected PCR directed restriction clone, but there's also other different methods that you can toggle or change between. Once you toggle between these different methods, under the hood, the software is actually applying the correct protocols to add, create primers for, say, Gibson cloning or infusion cloning, etc. Under the hood. So we're just going to keep it as PCR directed restriction clone. Under this vector tab option here, there's actually, if I click on this pull down, built into the software, there's actually cloning vectors that are already present. So we have a cloning vector catalog. So whether there's a number of different gateway vectors, common restriction vectors, M13 vectors, puck vectors, like so. You also have the capability or the option to bring in your own vectors and save to this catalog, or per a project, I can also import my own vectors and use them in a given project as well. So what we're going to do here is we're simply just going to right click on vectors. I'm going to import a vector of my own. So we'll just use P_4 dual and add that to a project. The next step, if I wanted to insert this into P_4 dual, is I'm actually going to click on this plasmid and drag it into, drag it into that vector. It's going to give me the approximate size of that plasmid with that insertion. The next step is I need to digest this. So what we're going to do is we're insert this and digest and specify what specific restriction enzymes we're going to use for that cloning. So what we've done is we just went from the target and now we're looking at the vector that we're going to insert into. On the bottom here, towards the bottom, hopefully everybody can see this, there's a yellow margin that gives you the option to specify the left and the right hand cut sites. Like so. By default, what the software is going to do is going to look for and apply single cutters that are present here in these pull downs. In this specific case, the two sites that we're going to utilize is actually NCO1 and KPN1 and the 3'. So this specific dual selection vector, it has some chloromphenical genes. There's some CDD and markers in which it can help with selection. We're simply just going to cut into the multiple cloning site between NCO1 and KPN1. So with that said, already working with this, I know there's actually more than one cut site for NCO1. So I'm actually going to click on this enzyme tab, type in NCO1, add that to the selectors that are being present, and then I'm simply just going to double click NCO5'. I'm sorry. And then click on KPN1 3'. Oh, it didn't update, sorry. There it is, from KPN1. Once you specify the appropriate cut sites, on the bottom right hand, like I mentioned previously in that details tab, it's going to be specific for what is selected. So in this specific case, I selected two different restriction enzymes. So it's going to show me the cut sites, the 5', top strand, bottom strand, cut sites for NCO1 and KPN1. The next step that you want to do here is actually select those two restriction sites. And then what that is going to do, it's going to take you back to the Untitled Cloning Project window. So this will give you a more accurate representation of that plasmid. So you're going to expect something around 4,501 base pairs in length for the final clone. The next step is I can click on the Summary tab. And then what this will do is it will actually give you the primers to add the appropriate ends to that target to be able to clone into that expression plasmid. One other additional thing here that is helpful is you can also look at the fragment junction, so the 5' to 3' end of the target, as well as the expression, the selection vector, and then you can actually see to make sure if things are in frame. If things look appropriate to you, the next step here is I would simply hit Make It, and then what the software is doing is creating the final restriction clone with those projects based on the project that you've specified. So here's my final clone. So under the feature tab, all the parental vector information is maintained, as well as the PCR product, restriction site, restriction product, the PCRs that are used for that strategy. And if I click on the primer tab of the PCR would be maintained, and this is something you can simply just file and export out as a primer catalog whether a FASTA file, a tab delimited file, etc. The next thing that I would do here is basically save this out. So I could simply file, save this out as a, whether a SeqBuilder Pro file, a standardized file format, a GBK, a FASTA file, EMBL, etc. that I can use for further downstream utilization. Another couple of things that might be helpful for you is, like I mentioned previously, built into the software, there is a feature library. If you wanted to send specific portions of this, maybe the restriction PCR project, to your feature library, this is where I can actually, under the feature pull down, send that to the feature library and then save that for further downstream work. So we have some customers doing that for genome work, antibody work, that if they come across or they worked with a specific nucleotide string of interest previously, and they want to very quickly search through their existing database, feature library database of information, you can very readily do so. All right, I'm going to close this out, and then the next step is basically taking this resulting restriction clone, and then we'll actually insert that into a gateway system. So I will take the clone that was created in that last step. There's my clone, like so. Under the cloning pull down, we're just going to do gateway expression clone. In this specific case, I'm going to import that clone again under the right click option. I'm going to right click on the vector folder and import my destination vector. So this is the PC DNA6 vector. The next logical step is I'm going to take the clone and add that to the pull down. Take that vector, add that to the vector pull down. It's going to give me the approximate size, as well as a summary, and then I can make it. And then what it's doing is using the recombination elements that are present, in that penter4 vector. It's going to correctly bring that into the final destination vector that you can kind of use for expression. So that would be basically the combination of using a restriction enzyme and then taking that restriction enzyme product and actually directly putting it into a gateway expression system. So these automated cloning, wizards are fairly new. I think we've started incorporating these probably 5 to 7 years ago, but literally it's easy as 1, 2, 3. In the first step, you're going to supply your insert. The second step is basically supply what vector that you want to insert it into with the appropriate ends and the primers that are automatically created for you. And then the final result is the resulting clone. And like I mentioned previously, all of those different in silico workflows would be present via the cloning pull down, like so. Okay. Any questions about cloning?
Yes,
Cloning
we have 2 cloning questions. Let's see here. So for restriction cloning, let me see, whether the software also adds the stuffer nucleotides in the primer, or we have to add them manually before ordering the primers. I'm not quite sure I understand that question. Okay, first of all, let's move on to another one for now.
Sorry. I can handle it. Yeah, okay. I can handle that question, Sharon. So yeah, that's a great question. So what we've done with the various cloning strategies that we support, whether it's PCR directed, gateway, TA cloning, Gibson infusion, what the software is automatically doing. So once I highlight a selection, it's automatically creating the primers for you. So once I've specified, the restriction sites that I want to clone into, or it doesn't necessarily have to be a restriction site, it simply could be a landmark or a location, in your expression plasmid. You just want to make sure that if it's in the multiple cloning site, obviously that it's in frame with whatever promoter elements that you're using for expression. So using the open reading frames, etc. But the software automatically creates those primers for you that you can, evaluate using that fragment junction window that I kind of show you. I can quickly jump to that for you here again. Click on sequence view, click on this selection here. Oh, sorry. Oh, it's not, I have to start from scratch, sorry. Yeah, so you simply go sequence view, click on your region of interest, clone, PCR directed cloning. So as you click through these various selections here, I'll just randomly specify a PGEM vector. We'll do PGEM 3Z, I digest. As you digest the vector, as you specify the left ends, right ends, please bear with me here. I'm just randomly clicking things, hit select. And then as you look at the summary window here again. So there's your primers. It's automatically creating those for you and then giving you fragment junctions to assess whether everything is in frame. Typically, like I mentioned previously, if you stay within the multiple cloning site, you're safe. But if you deviate outside the multiple cloning site, you may want to make sure that everything is in frame. That kind of segues into the next thing that we're going to kind of talk about in the software is the designer, the primer design functionality. So what we'll do is we'll open up that same that cow gene. So say you wanted to take this gene and do a more manual approach for primer design. You wanted to add restriction enzymes to the end of your target for whatever cloning strategy that you're utilizing. So under this tab, there's a primer design tab. So if I click on this primer design tab, it kind of shows me, potential primer binding sites, dimers, pair dimers, hairpins, etc. The easiest way to do a PCR primer design project is actually under the primer pull down. I would select create primer pairs. Once I select create primer pairs, it's going to give me the option to either add primers that end exactly at that selection, or the capability to choose an optimal primer location. So whether you're doing PCR primers or maybe you're doing sequencing primers, there's some nuances between the two. PCR primer pairs would be basically using the nearest neighbor method for calculations. In these primer characteristics, you can specify a target TM, unique setting at the 3' end, for specific how many base pairs. I can specify a minimum or maximum primer length, salt concentrations. If you want options, I can choose an optimal primer location I want to maybe stay within. If you're trying to find sequencing primers and maybe it's not as important to be at the beginning of a gene of interest, you just want something that hits somewhere within the gene, you can specify, stay within a given number of base pairs of that selection. Or if I want options, I can display at most however many different primer combinations or primer pairs. Once you've set those default characteristics or conditions, you simply just hit OK. And then what the software will do is actually create those primers for you across based on that criteria that I set. So in this specific case, it's giving me one result, a pair, and then it's giving me some results here in regards to the stable dimer information, the stable pair dimer, as well as hairpin formation. I can toggle between the top strand and the bottom strand primers. And that information would be updated so you can kind of visualize what's going on, across, that gene of interest, whether there's potential false binding sites of interest that could potentially exist. In regards to that restriction enzyme question, in here, there's also some various tools that you can kind of toggle between. The first one is simply just a selector. So it's a point and click, gives you the option to click things on, The second option is a scissor, which allows you to introduce a restriction site. So if I click on this scissor and say, here's the primer that's kind of created, this primer is also editable. So if I wanted to create a degenerate primer, I can highlight any of these base pairs and change it to whatever base pair that you would like. But say I may be interested in adding a specific enzyme of interest here at the 5' end, of this primer, or add the appropriate recognition sequence for that specific enzyme. So if I right click on that or I click on it, it's going to cut it and it's going to ask you, okay, what enzyme do you want to add? So it gives you all the different enzymes in your catalog that you have present, as well as the ability to, cut enzymes that don't cut the project. Or maybe you want to introduce, a stop mutation, etc., stop codon, etc. The other option here, as you create these primers and assess the stable dimer, pair dimer, or hairpin formation, you also have an option here to introduce a codon change or mutation. So say, for example, you want to click on some of these amino acids and see and incorporate a silent mutation to see if that potentially could make it a little bit more stable primer, or maybe you want to incorporate a non-silent mutation here directly with the primer. As you select some of these options, it will actually update the primer for you specific to whatever characteristic that you're changing there as well. So from here, as you create, edit, your primers are automatically saved to that primer view. It's going to give you your primer score, the length, of that product based on the 2 primers that are selected, TM, delta G, as well as you have the capability to file and save this out as a primer catalog. So whether as a FASTA file, a Lasergene primer catalog, or simply a tab delimited text that you want to send to your sequencing core for the primer creation. Any questions about primers? I know there was one question about whether you can, if you can do multiple sequences at a given moment. So from like a project and you want to, excuse me, do a more of a multiplex type of approach to primer design. Currently within software, there's not really a multiplex option. If you have 20 targets and you want to very rapidly or quickly create PCR primer combinations for 20 targets in silico all at once. Any questions about primer design?
Yeah, I think that you just answered one of them, which was, will the software design a bulk set of sequencing primers for a Sanger project? For example, pick primers every 500 base pairs for DS sequencing.
Yeah, so what you would do if you were creating like a scaffolding PCR primer pairs across the gene, you would select that the option stay within however many base pairs of the selection, and then you would want some flexibility in regards to creating alternate primers. Display at most, in this specific case, 30, 40, however many primers that you want to ensure an appropriate scaffolding across that gene of interest. So by default, the software, is set to create, the optimal PCR primer combination for an individual. But if you wanted different options or multiple primer combinations across the gene of interest, you would need to change these 2 settings under the create primer pairs option. So choose optimal primer location, specify how many base pairs of that selection, and then have some flexibility in regards to finding, alternate primers. If you're a current user, oh, sorry, go ahead.
Oh, Brian, so go ahead. You can finish up on this, and then I have one more question.
Yeah, if you're a current user and you have access to our SeqMan Pro application, there is actually the ability for primer walking, across this contig of interest directly in the software, where you can actually find multiple primers of interest, and that can kind of assist with some of the bulk primer options there.
Great. We have one question from Martin that came in right after the webinar started, and he's been waiting for an answer since then patiently. So I told him that you'd answer when you get to the primers part of the website. And that is, does SeqBuilder Pro have a function to screen a primer library for primers that align with a certain plasmid?
Good question. So built into that feature annotation tool, so under the feature pull down, the feature library manager, there's actually a number of different primers. So in that primer catalog, we'll kind of talk about that here in a second. There's a number of different parts from VecBase. So a lot of those are different common primers, M13 primers, T3, T7 primers. You also have the capability to actually bring in your own data. So if that data is primers, you can simply just add those primers into that feature library manager and then use it as a tool for auto annotation. when you bring in your data from whether NCBI or maybe sequencing data. And once you annotate the sequence, it will search through that built-in feature library manager for you. If you need assistance bringing in primers specifically, feel free to contact any of us here. The key thing is annotations. So the feature library would need to make sure that there's some type of annotations that are associated with that primer, but it's pretty easy to do that.
All right, so we have about 20 more questions that came in, but I wanted to let people know that since we're already right towards the end of the webinar here, we won't be able to answer those live, but what we are going to do is what we did for the Megaline Pro webinar a couple weeks ago. We're going to take all of your questions, both before and during the webinar. We will answer those and make an FAQ to put on our blog so you could see everybody's questions and the answers. And that is really helpful to me, especially to see those things, because I write the user guides and I will be able to incorporate those into future editions of the user guide. So it's really helpful to know what things people have questions about. Thank you very much. And we will send all of you an email in the next couple of days that has a link to the recorded webinar. And we will also send you another email in probably a week or so that has a link to the new FAQ that we're going to write. So that's all I needed to say. You can go ahead, Brian.
Awesome, thank you. I just want to say thank you everyone for your time today. I know we're pushing the hour here, but there's only a couple other things I would like to discuss, and one of those is codon optimization and automated cloning. So just want to say thank you for I know we're going a little over, but we're almost finished here. So what we're going to do here is we're going to bring in a protein. So I'm simply going to drag that from my folder again, bring in that protein of interest. So kind of like the nucleotides, you also have the capability to select file new protein. This protein is actually a xenoreductase, I believe from Old Yellow enzyme. So xenoreductases have the capability to degrade energetic compounds. When I was in graduate school, I actually did some nitroglycerin and TNT degradation using some research, using some different enzymes that could degrade those energetic compounds. So what we're going to kind of do is kind of discuss, the concept of codon, optimization. So within the software, if you're starting from the nucleotide, you currently have the capability to do a translation. Once you do that translation, there's actually going to be under the comments tab a breakdown of the different amino acids. So you could assess whether you potentially have rare codons. So what I'm doing here is, if you're starting from a protein and you simply just wanted to back translate into, say, a genetic code that has been optimized for E. coli expression, you can very easily actually do that within the software as well. So the first step that you'd want to do with that is under the sequence pull down, you have the capability under select genetic code to specify a given, specific genetic code that's present in the software. So by default, the standard genetic code is going to be selected. So if I would back translate this right now, it would give me that that ambiguity in the wobble position. So you don't want to do that. So if I wanted to express this protein in, say, an E. coli host of some sort, what I'm going to do is actually select the second one here, which is the strongly expressed non-degenerate E. coli back translation code. There's also a number of other different codes that is built into the software. You also have the capability to edit your own genetic code under this second option here, edit genetic code. But for today, what I'll do is I'll first set the strongly expressed non-degenerate back translation code. And then what I'm going to do is simply just highlight my protein of interest that I want to back translate. So once I highlight that protein, the next step is I'm simply just going to go to the sequence pull down, select new sequence, back translate. What the software does now is I simply, using that highly expressed E. coli back translated code, I have now optimized this specific nucleotide, this specific translation to be optimized for E. coli expression. The next step from here is now I could carry on in whatever expression system that I'm utilizing for insertion, and then basically with the understanding that now optimally I have nucleotide that it's ready for that specific host of interest. So commonly, if you're working with rare codons with translation, you might see translational stops or stalls, on your gels. But this is very helpful for people who are assessing that. As I scroll down here, again, it gives me those statistics as part of the back translation. So it's going to give me the molecular weight, the isoelectric point, the charge at pH 7 that you can further use for downstream analysis. The final thing I just wanted to kind of mention is a little bit more in-depth discussion of our feature library manager. So that would be present under our feature pull down, specifically feature library manager. Once I click on that, it opens up our library. I'm going to maximize this so you can see the whole thing. In the library, it shows you the annotated piece of information. So in this specific case, it's the M13 gene. There's any annotations that are associated with it, those would be populated and shown at the bottom here. These are all DNASTAR annotations that we pulled from VecBase. So on the bottom left-hand corner, if I click on DNASTAR, it shows me annotations from VecBase. The other option here in the bottom left, if I click on user, so these are all different user annotations that I've specifically added to this database per se. In here, you have the option to change the color, the style, the font, as you bring data into here. The final step to add data into this project, you would simply just highlight a selection. So, for example, this genome, I would drag and drop that into my database. It's going to pull all those features. You hit OK, and then the final thing you do is you basically hit X and then save out that database. To auto annotate, with the software, I would simply open up that. So I created a FASTA file just to show you. This is a coronavirus. I would drag that into the project or into the window here again. It's going to bring in that whole specific gene. If I click on the feature tab, there's no annotations that are attributed with this specific genome of interest. This is actually feline coronavirus, I believe. If I wanted to search for annotations, I would simply just click on feature pull down, annotate sequence, and an annotation parameter window is going to pop up. It's going to ask you whether you want to look for perfect matches, a minimum match percentage, minimum coverage percentage. I'll just use the defaults and select OK. It's going to open up that feature library. What it's doing right now is doing alignment or searching across that feature library for any potential annotations that fit my criteria. And then it's going to give me a results tab. In this specific case, most of these are perfect matches because I just dragged and dropped that entire genome, into my library. But in here, I can sort, if you brought every single possible coronavirus genome in here, you have the capability to sort by type, feature, depth of, the coverage, whether top strand, bottom strand, length. Once you basically identified maybe potentially some of these annotations of interest, the next step is you have the ability to accept these, reject these, maybe you want to replace them. And then the next step is to auto annotate by simply clicking on accept checked. What it's doing now is basically pulling those annotation, and then that FASTA file that we brought into the software has now become a annotated sequence file. If I click on the margin here and drag up, it will actually give you an annotation summary. So it gives you a timestamp, the name of the sequence or the project, as well as all those different annotations that are pulled and are auto annotated to your FASTA file of interest. It gives you the location as well as score information that this is something that you can further put in your laboratory notebook. So like I mentioned previously, when this was initially incorporated into the software, it was used as a plasmid annotation tool. But we have customers that are using this up at the bacterial form 5 megabase genome level for auto annotation.